Introduction. Welcome, students, to today's lecture on the fascinating topic of alanine metabolism. In this lecture, we will delve into the intricate details and mechanisms involved in the synthesis and regulation of alanine, a vital amino acid with diverse functions in our body. Through a comprehensive exploration of 12 key topics, we will gain a comprehensive understanding of the role of pyruvate transaminase, the intricate processes of alanine metabolism, and the implications of altered alanine metabolism in disease and various physiological conditions. So, let us embark on this scientific journey to unlock the mysteries of alanine metabolism. Pyruvate transaminase, or ALT, is essential for alanine synthesis. It transfers an amino group from glutamate to pyruvate, forming alanine. This reversible reaction occurs primarily in the liver, where it helps remove excess nitrogen from amino acids. By regulating ALT activity, the body maintains nitrogen balance and cellular homeostasis. Factors like hormones, substrate availability, and energy status influence ALT activity. This ensures a sufficient supply of alanine for physiological processes without disrupting metabolic equilibrium. Regulation of alanine metabolism involves hormonal control, substrate availability, and cellular energy status. Hormones like glucagon and insulin modulate enzyme activity. Glucagon stimulates gluconeogenesis, increasing pyruvate for alanine synthesis, while insulin promotes glucose uptake and glycolysis, reducing pyruvate and alanine synthesis. Substrate intake affects alanine production, with high amino acids increasing substrates and low amino acids limiting availability. Cellular energy status, indicated by ATP and ATP/ADP ratio, influences alanine metabolism. High ATP levels favor pyruvate conversion to alanine, while low ATP/ADP ratio shifts metabolism. These mechanisms ensure balanced amino acid supply and metabolic demands. The glucose-alanine cycle is a crucial metabolic pathway that links glucose and alanine metabolism. When muscles are under prolonged exercise or fasting, they break down amino acids, including alanine, which is then transported to the liver. In the liver, alanine is converted to pyruvate and can be used to generate glucose through gluconeogenesis. This glucose is released into the bloodstream for use by muscles and other tissues, completing the cycle. The glucose-alanine cycle helps maintain glucose levels and dispose of nitrogen efficiently, supporting proper metabolic function. One of the key functions of alanine metabolism is its role as a carrier of nitrogen. Nitrogen, derived from amino acids, needs to be efficiently transported and eliminated from the body to prevent toxicity. Alanine serves as a carrier of nitrogen by transporting it from peripheral tissues to the liver. As mentioned earlier, during periods of increased amino acid breakdown, such as exercise or fasting, skeletal muscle releases alanine into the bloodstream. Once inside the liver, alanine is deaminated, resulting in the release of ammonia and the formation of pyruvate. The ammonia produced from alanine metabolism is converted into urea through the urea cycle, ultimately being excreted in urine. This process ensures the safe elimination of excess nitrogen from the body. By acting as a nitrogen carrier, alanine metabolism contributes to maintaining nitrogen balance, preventing nitrogen toxicity, and supporting overall metabolic health. Alanine metabolism differs between tissues due to varying metabolic needs. In the liver, alanine is used for gluconeogenesis and regulation of blood glucose levels. Skeletal muscle releases alanine during catabolism, providing a means to transport excess nitrogen to the liver. The heart and kidneys also engage in alanine metabolism based on their metabolic demands. These tissue-specific differences contribute to the overall maintenance of metabolic homeostasis. The efficient translocation of alanine across cellular membranes is pivotal for its role in nitrogen transport and overall metabolic function. This process involves specific transport systems to ensure the uptake and release of alanine as needed. In peripheral tissues, such as skeletal muscle, the plasma membrane exhibits specialized transporters known as alanine aminotransferase, ALAT1 and ALAT2. These transporters facilitate the uptake of extracellular alanine into the cells for subsequent breakdown or utilization in metabolic processes. In the liver, alanine is transported into hepatocytes through specific membrane transporters. Once inside the liver, alanine is readily converted into pyruvate through transamination reactions, as discussed earlier. The precise mechanisms underlying alanine translocation across cellular membranes are still being investigated. However, the existence and activity of specific transporters highlight the importance of maintaining a tightly regulated transport system to ensure proper alanine metabolism and metabolic homeostasis. Pyridoxal phosphate, PLP, the active form of vitamin B6, functions as a crucial cofactor in alanine metabolism. PLP participates in transamination reactions, facilitating the transfer of amino groups between various amino acids and their corresponding keto acids. In the context of alanine metabolism, PLP serves as a cofactor for pyruvate transaminase, which catalyzes the conversion of pyruvate and an amino acid, usually glutamate, into alanine and another keto acid. This reaction is reversible, allowing the interconversion of alanine and pyruvate based on the metabolic needs of the body. The role of PLP in alanine metabolism highlights the intricate interplay between vitamins, cofactors, and enzymatic reactions. Deficiencies in vitamin B6 can impair alanine metabolism and disrupt nitrogen balance, emphasizing the importance of adequate vitamin intake for optimal metabolic function. Altered alanine metabolism can have significant implications for various diseases and pathological conditions. Dysregulation of alanine metabolism may contribute to metabolic disorders, liver diseases, and disturbances in nitrogen balance. Liver diseases, such as hepatitis or cirrhosis, can lead to impaired alanine metabolism. This can result in elevated blood alanine levels, as the liver's capacity to efficiently utilize or dispose of alanine may be compromised. Elevated alanine levels in the blood can serve as a diagnostic marker for liver dysfunction. Additionally, disruptions in alanine metabolism can impact nitrogen balance and lead to nitrogen toxicity. Certain inborn errors of metabolism, such as hyperammonemia, can arise from defects in enzymes involved in alanine metabolism, leading to impaired nitrogen disposal and resulting in ammonia accumulation. Understanding the disease implications of altered alanine metabolism aids in the development of diagnostic strategies, therapeutic interventions, and management approaches for individuals affected by such conditions. The metabolic demands during exercise and fasting impose unique challenges on alanine metabolism. These physiological conditions induce alterations in alanine metabolism to meet the energy requirements of the body while maintaining nitrogen balance. During exercise, skeletal muscle catabolism increases, leading to the release of alanine into the bloodstream. The released alanine is utilized by the liver for gluconeogenesis, contributing to the maintenance of blood glucose levels for energy production. Fasting also triggers changes in alanine metabolism. As the body depletes glycogen stores, alanine released from skeletal muscle serves as a substrate for gluconeogenesis in the liver. This process helps maintain glucose homeostasis and provides an alternative energy source when carbohydrates are limited. The adaptations in alanine metabolism during exercise and fasting are essential for energy supply, maintenance of blood glucose levels, and efficient nitrogen disposal, ensuring metabolic flexibility under diverse physiological conditions. Alanine metabolism is intricately interconnected with various other amino acid pathways, highlighting the complex network of amino acid metabolism. These interconnections allow for the synthesis, breakdown, and conversion of amino acids to meet the diverse metabolic demands of the body. One notable interconnection is the relationship between alanine and glutamate. Alanine transamination, catalyzed by pyruvate transaminase, utilizes glutamate as the amino group donor. Conversely, glutamate can be regenerated from alanine through the reverse transamination reaction. Another vital interconnection is between alanine and the branched-chain amino acids, BCAAs, which include leucine, isoleucine, and valine. Alanine serves as a carrier of nitrogen for the BCAAs during their breakdown, aiding in the removal of excess nitrogen and maintaining nitrogen balance. These interconnections exemplify the intricate web of amino acid metabolism, showcasing the dynamic interactions between various pathways to regulate and support metabolic processes. In the field of alanine metabolism, transamination reactions play a vital role. These reactions involve the transfer of amino groups between amino acids and keto acids. Pyruvate transaminase, an aminotransferase enzyme, is specifically involved in alanine metabolism. These enzymes rely on pyridoxal phosphate, PLP, as a cofactor for their activity. The molecular mechanisms of transamination reactions involve the formation of a Schiff base intermediate between PLP and the keto acid. This intermediate enables the transfer of the amino group from the donor amino acid to the keto acid, resulting in the formation of the corresponding amino acid and another keto acid. The selectivity of transamination reactions is determined by specific binding and recognition sites within aminotransferase enzymes. Understanding these molecular mechanisms enhances our comprehension of the precise control and coordination of alanine metabolism. In summary, the allosteric regulation of enzymes involved in alanine metabolism allows for rapid responses to metabolic demands. Examples include the allosteric regulation of pyruvate kinase by metabolites such as fructose 1,6-bisphosphate and ATP, which ensures the availability of pyruvate for alanine synthesis. Additionally, alanine aminotransferase is feedback inhibited by alanine itself to prevent excessive production. These regulatory mechanisms maintain metabolic balance and enable optimal metabolic function in the body. In conclusion, understanding the complexities of alanine metabolism is crucial for maintaining metabolic balance and overall health. We explored the synthesis of alanine, regulatory mechanisms, the glucose-alanine cycle, and tissue-specific differences. We also discussed the role of alanine in nitrogen disposal, translocation across membranes, disease implications, and metabolic adaptations during exercise and fasting. Additionally, we examined the interconnections with other amino acid pathways, transamination reactions, and enzyme regulation. This knowledge helps us understand the biochemical processes and the impact of altered alanine metabolism in different conditions.